Okay, so we are the Kanha group, and our project is the effects of RG108, a human DNA methylation enzyme inhibitor, and its related analogs on HCT116 colorectal cancer cells. In our presentation, we'll be going over an introduction to RG108 and our research, a description of the methods we utilize in our research, a brief overview of our current results, future assays that we plan on doing, and finally our conclusion and references.
To begin, here's some background on our research. So colorectal cancer, or CRC, is a development of cancer in the colon or rectum, and it is the second most common cancer leading to death. Epigenetic therapy is a new cancer treatment method that displays promising clinical data in the treatment of colorectal cancer, leukemia, lung cancer, and more. Epigenetic therapy is the use of drugs and other epigenome-influencing techniques to regulate the activation and inhibition of certain genes without alteration of nucleotide sequence. Epigenetics involves DNA methylation and modification of histone proteins and microRNA that consequently affect gene expression. In its potential for cancer treatment, epigenetic modifiers may activate genes that allow for apoptosis through demethylating certain regions of the DNA, as well as promoting the methylation of oncogenic genes to inhibit their expression in cancer cells. Certain tumor-suppressor genes were chosen based on their antiproliferative traits. These genes work to slow down cell cycle progression and induce apoptosis through anti-angiogenesis or regulating myogenesis. N-Phthaloyl or RG108 is a DNA Methyltransferase 1 DNMT1 inhibitor. It is a non-nucleoside inhibitor that, rather than getting incorporated into the DNA, inhibits the active sites of DNMTs, which helps mitigate its toxicity. RG108 has potential in its high specificity for DNMTs, low toxicity, and has been shown to significantly reduce methylation in human colon cancer cells. Our interest in RG108 and its analogs is initiated by previous studies focused on DNMT inhibition by nucleoside and non-nucleoside inhibitors. Currently, only two DNMT inhibitors are FDA-approved, azacitidine and decitabine. As a pyrimidine nucleoside inhibitor, azacitidine works to inhibit DNMT while reactivating tumor-suppressor genes. Another nucleoside-type DNMT inhibitor, decitabine, works similarly, leading to DNMT depletion and genome hypomethylation. As DNMT can also be inhibited by non-nucleoside inhibitors, hydrolyzine and procainamide work to reactivate tumor-suppressor genes in breast cancer. In previous studies, the nucleoside inhibitors, azacitidine and decitabine, have been more effective at inhibition when compared to the non-nucleoside inhibitors, hydrolyzine and procainamide. The following research studies additional analogs of RG108 to potentially compare its efficacy with nucleoside inhibitors by focusing on the direct effects of RG108 and its analogs on the reactivation of tumor-suppressor genes and selectivity. Our experimentation also utilizes imidazole, an aromatic diazole and alkaloid, working to interact with DNA via covalent or non-covalent interactions and halt cell division. The molecule does not work like the inhibitors previously described and instead binds to proteins to inhibit cancer cell division. Nonetheless, our work utilizes imidazole as a control point, considering its strong anti-cancer properties as a comparison for the efficacy of novel RG108 analogs. For the purpose of this experimentation, human colorectal carcinoma 116 or HCT116 cells were used. The cells initiated by an adult male are adherent with an epithelial morphology, meaning that they are polygonal in shape and adhere to a substrate. When implanted into immunocompromised mice, the cells form primary tumors and distant metastases. The cells can be tracked in vivo, making them viable tools for studying tumor growth mechanisms and evaluating the effects of drugs and therapies. Our experimentation aimed to locate correlations between decreased cell viability and introduction of RG108. In order to test the efficacy of RG108 analogs, we utilized various experimentation methods. MTT assays will be applied to measure cellular metabolic activity, indicating cell viability, cytotoxicity, and proliferation. Our work will also implement genetic technology with quantitative polymerase chain reaction assays, or qPCR, with the extraction of RNA to quantitate alterations in gene expression connected to the revival of tumor suppressor genes. Given that RG108 is a DNA methyltransferase inhibitor, sections of methylated DNA will be explored in order to test its efficacy.
We maintain the HCT116 cell line through consistent splitting in order to keep our cells healthy and prepared for experimentation. Every 2 to 3 days, when cells were conflated, media was aspirated off of the flasks carefully, and 1 milliliter of Delbecco's phosphate buffered saline was pipetted into the flasks. Next, DPBS rinse was repeated, and 1 milliliter of 0.25% trypsin EDTA solution was pipetted into the flasks. The plates were incubated at 37 degrees Celsius for 5 minutes, and the trypsin reaction was neutralized as the cells were transferred into a new flask with 9 milliliters of McCoy's 5A medium with 10% fetal bovine serum and 1% penstrep solution. Cells were frozen and later thawed in order to keep uniform, healthy cells in preparation of experimentation. Cells were frozen by centrifuging them in the correct amounts of media, and a pellet was observed. Once replacing the media, the pellet was resuspended and moved to cryovials. These were kept frozen in the negative 80-degree freezer. Cells were occasionally thawed based on our needs for experimentation. Approximately 70% of the cells were thawed and added to centrifuge tubes filled with media. Similar to the freezing procedure, the tubes were centrifuged to obtain a pellet and replaced with media. They were then added into flasks to commence with regular splitting. We completed a cell proliferation assay in order to assess our cells' ability to divide in response to different stimuli. We suspended the HCT116 cells in a trypsin solution and seeded them into a 96-well plate. After a 24-hour incubation, we added the appropriate drug concentrations to each column. Column 1 contained cells and regular media, column 2 DMSO, column 3 100 micromoles of RG108, column 4 250 micromoles of RG108, column 5 500 micromoles RG108, and column 6 24 micromoles imidazole. After a 72-hour incubation period, at 37 degrees Celsius, the cells in each well were counted with the use of the Countess automated cell counter. The MTT assay was used to test cell viability and therefore the efficacy of another anti-cancer drug, imidazole, and RG108 against tumor growth of HCT116 cells. Once suspended with trypsin solution, HCT116 cells were first plated into a 96-well plate with proper cell concentrations. After 24 hours of incubation at 36 degrees Celsius for cell adherence, the cells were treated with the different drug concentrations of imidazole and RG108. The drug concentrations were incorporated with the following amounts: row A, 24 micromolars, imidazole; row B, 500 micromolars, RG108; row C, 1000 micromoles, RG108. Next, 50 micrograms of thiazole blue tetrazolium bromide and 10 milliliters DPBS solution was pipetted into the wells and incubated for 4 hours at 36 degrees Celsius. 80 microliters of DMSO was added into the wells and incubated for 15 minutes at 36 degrees Celsius. The plate was then run under a plate reader, and results were determined with the raw data received. Similarly to the MTT assay, the Resizurin assay was used in order to test cell viability and drug efficacy. The cells were suspended in a trypsin solution and seeded onto a 96-well plate. They were incubated for 24 hours at 37 degrees Celsius before the appropriate drug concentrations were added to half the wells. In row A, there was a regular media, row B, DMSO media, row C, 100 micromoles RG108, row D, 250 micromoles RG108, and row E, 500 micromoles RG108. After another 24 hours, the appropriate drug concentrations were added into the remaining wells in the same order as before. Then 500 micrograms of Alamar Blue and 10 microliters DPBS solution was pipetted into the wells and incubated for 4 hours at 37 degrees Celsius. Then 80 microliters of DMSO was added into the wells and incubated for another 15 minutes at 37 degrees. The plate was then run under a plate reader, and the results were determined with the data received. We also completed RNA extraction and preparation of qPCR in order to isolate and analyze genes of interest. The HCT116 cells were treated with the drug concentrations and incubated for 72 hours. 3 flasks were treated with 24 micromoles of imidazole, 3 with 500 micromoles of RG108, and 3 with 500 micromoles of DMSO. After the incubation period, the New England Biolabs RNA extraction kit was used to lyse the cells and extract the DNA. Cells were then kept on ice and stored at negative 80 degrees Celsius in RNase-free cryovials in order to preserve the RNA. The mRNA was then analyzed with the use of the nanodrop. A clonogenic growth assay will be used in order to determine cells' reproductive levels after treatment with RG108, imidazole, and DMSO. The HCT116 cells were treated with the drug concentrations of 24 micromoles imidazole, 500 micromoles RG108, and 500 micromoles DMSO. The cells will then be incubated at 37 degrees for 14 days with regular media replacement every 2 to 3 days alongside cell maintenance. After 2 weeks, the media will be removed from the plates and they will be washed with 0.9% saline. The colonies will then be fixed with a 10% formalin solution for 30 minutes, after which excess crystal violet will be washed off with distilled water and the colony growth will be measured with pictures taken by a microscope, and digital images will be obtained and analyzed with the use of ImageJ. A protein extraction was also completed in order to prepare to analyze various proteins of interest. The cells were rinsed with DPBS, trypsinized, and then centrifuged in order to count them to determine lysis buffer volume. The cells were then suspended in lysis buffer and lysed. The solution was then centrifuged for 30 minutes in the microcentrifuge at 13,000 RPM at 4 degrees Celsius. Afterwards, they were kept in ice and stored at negative 80 degrees Celsius in order to preserve the protein. A Bradford assay was also completed in order to measure the amount of protein in a sample and help predict cell activity. Bradford reagent was added to 14 microtubes, and a BSA standard was created with Bradford reagent in the following concentrations: 0 micrograms per microliter, 5 micrograms per 30 microliters, 10 micrograms per 30 microliters, 15 micrograms per 30 microliters, 20 micrograms per 30 microliters, 25 micrograms per 30 microliters, and 30 micrograms per 30 microliters. Our protein sample was then mixed with Bradford reagent and the absorbance was measured. Afterwards, a standard curve was created with the resulting data. DNA gel electrophoresis will be conducted in order to analyze target segments of DNA. The DNA will be isolated with the use of a DNA extraction kit. Then the preserved DNA will be added into an agarose gel alongside a DNA ladder, and the sample will be electrophoresed for 40 minutes at 50 V, and the results will be analyzed.
I will now be talking about our results, which mainly include MTT assays. The purpose of this experiment was to validate our MTT assay technique by comparing our results with the results of a previous paper. We used the drug imidazole, an aromatic heterocycle, which has been shown to activate apoptotic pathways in HCT116 cells. We compared our MTT results with the results of the paper to validate our technique. The figure shows the cytotoxicity of imidazole in HCT116 over T equals 0 hours and T equals 24 hours. The percent of cells dead was graphed across the different drug concentrations. 3 million cells per valve were seated in a 96 valve plate. The cells were treated with imidazole at the stated time points. After incubation with the MTT dye and solubilization of formazine with DMSO, absorbance was measured at 570 nanometers and used to calculate cytotoxicity values. The error bars are the standard deviation. The purpose of this experiment was to compare the effects of different drugs on cell viability. Imidazole, decitabine, and RG108 were used in this assay. We added decitabine, an FDA-approved nucleoside analog DNA methyltransferase inhibitor, which induces DNA demethylation. As decitabine is both a DNMT inhibitor, which is the same class of drug as RG108, and also already FDA-approved, we hope to use decitabine as a comparison point to further validate our results. This figure shows cell viability of HCT116 cells treated with 100 micromoles of imidazole, decitabine, and RG108 across T equals 0 hours, T equals 24 hours, T equals 48 hours, and T equals 72 hours. 3 million cells per milliliter were seated in a 96 valve plate. After drug treatment, incubation with MTT dye, and solubilization of formazine with DMSO, absorbance was measured at 570 nanometers and used to calculate cell viability. The error bars are the standard deviation. The purpose of this experiment was to compare the effects of different concentrations of RG108 on cell viability. This figure shows cell viability of HCT116 cells treated with 100 micromoles, 250 micromoles, and 500 micromoles of RG108 over T equals 24 hours. 3 million cells per milliliter were seated in a 96 valve plate. After drug treatment, incubation with the MTT dye, and solubilization of formazine with DMSO, absorbance was measured at 570 nanometers and used to calculate cell viability, and the error bars are standard deviations. The purpose of this experiment was to compare the effects of different concentrations of RG108 across different points on cell viability. This figure shows cell viability of HCT116 cells treated with 100 micromoles of imidazole, 100 micromoles of DMSO, and 100 micromoles, 250 micromoles, and 500 micromoles of RG108 over T equals 0 and T equals 24 hours. 3 million cells per milliliter were seated in a 96 valve plate. After drug treatment, incubation with the MTT dye, and solubilization of formazine with DMSO, absorbance was measured at 570 nanometers and used to calculate cell viability. I will now discuss the results. Overall, our MTT assay results were generally inconsistent with large margins of error. For example, an increase in cell viability with time was observed when treated with increased RG108 concentration. This was observed in the 250 micromole treatment, which is on slide 21, comparing cell viability values from the time points of 0 hours and 48 hours. This was also observed in the cell viability of cells treated with 150 micromoles, 250 micromoles, and 500 micromoles of RG108. There was also an increase of cell viability with the cells treated with 500 micromoles of RG108, while the cell viabilities for cells treated with 150 micromoles and 250 micromoles was mostly the same. On the other hand, treatment of our cells with DMSO, which was meant to be the negative control resulting in complete cell death, didn't change the measure to cell viability. Because of these inconsistent results, we decided to switch to the Alamar Blue assay. The Alamar Blue assay involves less manipulation steps than the MTT assay, which means it has less opportunities for error. We are now currently working on optimizing the Alamar Blue assay and RNA and protein extraction.
During our research, we experienced many challenges such as maintaining cell lines. We experienced initial problems with maintaining our cell lines as we ran into problems such as dead cells and low cell count due to mistakes in the procedure. But with lots of practice, we were able to successfully sustain our cell lines and even freeze many for later. Another challenge that we faced was counting cells with a Countess cell counter. Our first time counting cells with the machine gave us really low viability levels, but after going over the protocol for the machine and using the right settings, we had very high viability with recounted cells. We also had to optimize RNA extraction. We used one kit and different tubes to make sure our quantified RNA gave us satisfactory results, and in the future, we plan on using new kits as well. Furthermore, after an unsuccessful MPT, we considered the amount of cells being plated into each well as one of the contributing factors. To ensure each well had the same amount of cells, we plated our cells for 24 hours and chose 3 time points, a 48-hour time point, a 72-hour time point, and a 96-hour time point to count them. After achieving successful results, but still similar unsuccessful results in our MTT, we decided to switch to the Alamar Blue assay, which had less manipulation steps compared to the MTT. This would result in less technical errors, and as of now, we are currently optimizing the Alamar Blue assay. In the realm of cancer biology, there are certain experiments showing the different characteristics of cancer cells. Here are some of the assays that we plan on running for different purposes. We'll be using qPCR to analyze the reactivation of tumor-suppressor genes when treated with RG108. qPCR will allow us to see the number of repetitions of DNA duplication needed for a fluorescent signal to be visible. With immunoblotting, we'll be analyzing tumor-suppressor proteins before and after RG108 treatment. Immunoblotting will be used by first sorting proteins through gel electrophoresis, then they will adhere to membranes, also known as blotting, where they are then accessible for detection using antibody binding. We'll be using flow cytometry to monitor cell apoptosis, as a flow cytometer machine analyzes physical and chemical characteristics of cells. The clonogenic growth assay will help us analyze the ability of cells to form colonies under different conditions. Cells plated and growing will be treated with RG108 to see the number of colonies in untreated and treated cell wells. The number of colonies are then counted by staining cells with crystal violet. Next, we have fluorescent microscopy, which uses fluorescence that lets us analyze cells. And finally, the wound healing assay will analyze the cell's ability to migrate. After conducting the validation experiments mentioned above, future work aims to specialize in the treatment of novel RG108 analogs and reactivation of tumor suppressor genes. We will also be determining whether the analog's capability for immunomodulation would have increased efficacy when compared with RG108 and other FDA-approved drugs. And for our conclusion, so here's a quick overview of all our work. We started off with cancer biology literature reading about new developments with colon cancer cells and DNMT inhibitors. We then maintained our HCT116 cell lines with cell splitting, freezing, and thawing. Throughout this process, we continued our literature readings and also worked towards starting to perform experiments with our cells. We first read about an assay that we were going to conduct, created a protocol with it, and then conducted the experiment. This process occurred with several assays, including a cell proliferation assay, the clonogenic assay, our MTT assay, RNA extraction, the Bradford assay, and the Alamar Blue assay. In the future, we also hope to learn R programming, which will help us compute our data. Essentially, we are testing analogs of RG108 as new non-nucleoside DNMT1 inhibitors for improved efficacy in treatment against tumor growth and revitalization of tumor suppressor genes. We plan to continue our research with the implementation of genetic technology with use of Western blotting, gel electrophoresis, and various cell assays. As we proceed, we expect that our research will reveal diverse treatments for colorectal cancer through DNA methyltransferase inhibition. As of now, we are optimizing current assays for pure RNA from qPCR, clear Alamar Blue results, and protein from future Western blots. Reliable results with RG108 will give us a control that we can use to compare RG108 analogs, which we hope to have more success with. Our work aspires to evolve into FDA-approved drugs in the future, following a pathway similar to that of azacitidine and decitabine. Overall, we aspire to confirm the efficacy of RG108 and its analogs on the reactivation of tumor suppressor genes and selectivity. Here are our references.
We would like to thank Mr. Clinton Cunha for being a wonderful advisor, inspiring our research, and providing us with the resources to succeed. Thank you. Mr. Edward Enjo for providing us with RG108 and RG108 analogs to experiment with, thus fueling our entire experimentation. Thank you to the lab techs at ASDRP for ensuring our safety and well-being throughout our work and supplying us with the materials we required. Finally, thank you to the Aspiring Scholars Directed Research Program and the Olive Children Foundation for providing us with the resources and opportunity to conduct this research.
Thank you for listening. And if you would like to take a closer look at our references, please feel free to scan the barcode.
Okay, so again, that question rises that if I pick 3 participants right now in Colloquia, how many of them can summarize your research study?
Sahar, I think the best way to test that scientific question is to pick 3 people at random from the Colloquium audience.
Yeah, that I think that's what I'm gonna do.
Oh, okay. You go.
Vedant Gupta. Vedant Gupta, can you hear me?
Oh, people are leaving like this. We're not.
Okay, Manisha's iPhone. Anushri. Okay, you know what? Who are your audience? High school students, just like you, right? This presentation was way too advanced of even undergraduate degree. I wish I could bring questions, which I do have one, but I was just listening because it was just a different audio going through some reading material and reading it fast. It was hard for me to catch up on a slide. It was just like very obvious you guys prepare a couple of, reading things that you were been just reading and going through. So I would recommend you come back again to this Colloquia and present it in such a way that every single one in this Colloquia understand what you have actually done. Because a work is valuable unless when you hear it, you understand what's going on. You can do the best thing in the world, but if no one really understands in a very simple way what you have done, it's a little bit confusing. I appreciate that conclusion slide at the end, but it was, again, you guys talk so fast, I was keep on, losing the track. Like clonogenic growth assay versus Bradford assay. Like if you want to bring all those things, you have to maybe break your presentation to a smaller one, and each one of you take a charge of section of it and present it in different Colloquia. And can we go to page 20? Okay. Maybe a slide after that, maybe, or before or after 21, 22, maybe it was one graph that it was confusing for me. Not this. Okay, never mind. Maybe I misunderstood. It's okay. Yeah, forget about it. Yeah, I wish I could, I wish I had some question to ask, but yeah, it was hard for me to track.
I have a question.
Come back again.
I have a question, Sahar, if you don't mind. So there was, I think you're using what, imidazole as a comparative agent compared to RG108. Is that, am I correct in that?
Yeah.
Why imidazole? What's the toxicity? What's the LD50 of imidazole?
So I don't have like specific LD50 values off the top of my head, but we initially decided to just use imidazole just as when we were first running our MTT assay, we wanted to make sure we were running it right. And imidazole had results, MTT results with the same cell line that we were using. So we were using that.
Why imidazole? Imidazole is used as a buffer, right? This thing is pretty non-toxic, especially in cancer cells. You could feed a mouse. The LD50 is just under 1 gram per kilogram. Okay, you could feed a mouse like 0.1% of its weight of imidazole, and it wouldn't die. It's like, it's not quite sugar, but it's not a very toxic material, and certainly not a cancer drug. So I'm curious to know what made you pick imidazole over the hundreds upon thousands of other compounds out there. And there's a lot of things, by the way, that are just toxic, right? If I dump lead acetate is one of the best anti-cancer agents because it's going to kill every single cancer cell. It's also going to kill everything else. Same thing with methyl iodide. So why imidazole?
Because we did find a couple of papers that did show that imidazole had like inhibited.
Can you reference one of those papers?
Yeah, I can actually put it in the chat right now. For the cell line we were using, and we just kind of simply used it in the beginning, just kind of as since it already had provided MTT assay results, we just wanted to compare whatever results. So it was kind of more of a validation experiment.
Okay, but I still don't understand why you're using imidazole as a validation experiment. Like there's hundreds upon thousands of other compounds out there that kill cancer cells exceptionally well. And I don't know if this is reproducible because this is not consistent with other literature protocols.
I think while that's accurate, we really just picked imidazole because we found papers that used very similar protocols to what we are using. So for instance, specifically MTT assays and flow cytometry.
But everybody does MTT, right? I mean, I'm going to do an MTT in about 30 minutes here. So, everybody does. And MTT is like one of the most common viability cell proliferation assays that is performed, as is the blue one. So, but that doesn't answer the question of why you're using imidazole.
Yeah, well, of course, like that's like very true.
So RG108 is a DNA methyltransferase inhibitor, right?
Mm-hmm.
Does imidazole do the same thing?
Imidazole works completely differently.
Yeah, and that's why like added decitabine, because that one actually has the same kind of drug class as comparison point. But initially we went with imidazole because, well, that was like one of the papers we had found.
How does an imidazole induce cell death?
It interacts with one of the apoptotic proteins.
It binds to proteins and it halts cell division from there.
Okay, but there's a lot of things that bind proteins, right? Sugar binds proteins.
It like promotes activation of caspases. It promotes pro-apoptotic protein expression, and
I would recommend that you all give some thought to. I mean, we have a library of anti-cancer drugs that one could pull from, right? If you say your compound works better than imidazole, that's like saying, oh, well, it works better than salt. That's not saying a whole lot.
And yeah, that's why we eventually.
I can't find a single other literature reference that cites the anti-cancer activity of imidazole except for this paper which you've referenced, which comes from a journal of questionable quality. Just because something is posted on the NCB or on the NIH website, I mean, there's a lot of journals that are indexed there. And the Medical Science Monitor, what's the impact factor of that journal? So.
Yeah, that's a completely valid concern. That's why we future like implemented decitabine later on. Keeping that in mind, we probably will continue to use RG108 and compare it to like azacitidine, potentially also comparing it to its non-nucleoside inhibitor, like that, like inhibitors like hydroxyzine and procainamide potentially. So yeah, we really just use imidazole as a starting point. Looking back now, I think we can really move forward using some more tested drugs in that sense, but yeah.
And then a second question. What you mentioned qPCR as one of the techniques. That's a very powerful technique, and as many of you know, that has been used extensively for SARS-CoV-2 testing in recent years. What genes are you planning on studying through PCR?
So we're planning on studying tumor-suppressor genes. So I believe one is like P16, another one is like a transcription factor gene. These all have been shown to be methylated in colon cancer cells. So and previous
Are they methylated in regular cells?
Sorry.
Are they methylated in regular cells?
No.
They're not.
So, and RG108 has already been shown to demethylate these specific genes. So we are first.
Does it demethylate it, or does it inhibit the enzyme that methylates it?
Sorry, it inhibits that enzyme that methylates it. Yeah, and it causes demethylation. So yeah, we're going to hopefully study that, and then hopefully start like expanding, because right now we're just looking at the genes which have previously been validated with previous RG108 papers.
Okay, and if since the RG108 I made that compound for you guys, but since that comes from ASDRP, it's synthetic compounds. We have the potential to make other analogs. What analogs would you propose and why?
I think we were thinking of doing I believe a 5. I'm not sure the exact name, but it was like a 5-HTT compound, and
5H.
Fluoro, yeah, 6-fluoroinidol version, a 6-fluoroinidol analog in a 5-hydroxy. So I think like we had done a bit of background, and we found that 6-fluoroinidol had like some immunomodulatory properties. So that's why we had decided to.
Say indazole? Are we putting indazoles or indoles? If you had an indazole, would it be better or worse? Okay, so I encourage you to give that some thought, because these compounds can be produced at will, and that that's powerful for your drug discovery effort. Okay. Good presentation.
Hey, thank you.
Good work, Lynn. This is we got kids doing MTT assays. That's awesome.